Today we will talk about the test cross. What the test cross is, what is the objective of the test cross, why it is being performed. This test cross is simply a cross between individual with dominant phenotype and individual with recessive phenotype. So we can say that it is a cross between individual with dominant phenotype and individual with recessive phenotype and individual with recessive phenotype So what do we mean by test cross? Test cross is simply a cross between individual with dominant phenotype and individual with recessive phenotype. Or we can see also we can say that, in other words, we can put it like this: in test cross, a cross is made between the individual with dominant phenotype with the homozygous recessive individual. So here test cross is simply individual with dominant phenotype it is crossed with homozygous recessive parent, homozygous recessive individual. So what is this test cross, where in test cross the individual with dominant phenotype is crossed with homozygous recessive individual. This is what is called as test cross. The individual with dominant phenotype is crossed with homozygous recessive individual. What is the objective of test cross? Objective of TC, test cross. Why test cross is being performed? We perform the test cross so as to determine the individual with dominant phenotype, whether it is homozygous or heterozygous for the trait. We have got this individual with dominant phenotype. We don't know the genotype of this individual, whether it is homozygous or heterozygous. So in order to determine the individual with dominant phenotype, whether it is homozygous or heterozygous for the trait, we perform test cross. So objective of test cross is to determine whether individual with dominant phenotype, individual with dominant phenotype is homozygous or heterozygous for the trait, So in order to determine whether the individual with dominant phenotype it is homozygous or heterozygous for the trait, we perform test cross. Suppose in case the individual with dominant phenotype is homozygous. Suppose we have individual with dominant phenotype individual we have this dominant phenotype individual we want to determine whether this dominant phenotype individual, whether it is homozygous or whether it is heterozygous for the trait or it is homozygous for the trait. That means the dominant trait, it has got a dominant trait, but we don't know whether this dominant is homozygous or heterozygous. Dominant phenotype individual, in case the dominant phenotype individual is first we'll take homozygous. So we have case first. That means dominant phenotype individual can be homozygous. If it is homozygous, that means homozygous means it has two identical alleles for the trait. It has two similar alleles for the trait. For example, capital T, it is homozygous. Capital R, it is homozygous. It is homozygous condition. Whether it is, then all the offsprings, all the progenies which are produced when this dominant phenotype individual is crossed with the homozygous recessive parent, with the homozygous recessive individual, then in F1 generation all the progeny or all the offsprings would have dominant trait. That means in F1 generation all progeny, now we can say that all offsprings would have dominant trait. Would have dominant trait. If this dominant phenotype individual is homozygous and then it is crossed with the homozygous recessive individual, then in the F1 generation we will get the offspring, we will get the progeny, they would be all having the dominant trait. There would be no recessive trait in the F1 generation in case the individual with dominant phenotype is homozygous. Then case second, if individually dominant phenotype individual, if this dominant phenotype individual is what? It is heterozygous for the trait. Heterozygous. If it is heterozygous, that means there are two dissimilar alleles for the trait. Its heterozygous would be like this, capital T, small t, or capital R, small r. This is heterozygous condition. And then it is being crossed with the homozygous recessive individual. Then in F1 generation, we will get 50% with dominant trait and 50% with recessive trait. That means there would be a progeny, the ratio of progeny would be 50% offsprings with dominant trait and 50% offsprings with recessive trait. Offspring is with dominant trait and 50% offsprings would be having the recessive trait. That means the ratio of the progeny or the ratio of the offsprings in case of test cross ratio would be 1:1. That means the ratio of offsprings or the ratio of progeny, ratio of offsprings or the ratio of progeny in case of test cross would be 1:1. That means 50% would be having dominant trait, 50% of the progeny would be having dominant trait, and 50% of the progeny or 50% of the offsprings would have recessive trait. That means in case of if the individual is, now we have got two cases. First case is we have got an individual with dominant phenotype. If it is homozygous, then in the F1 generation all the individuals would be having the dominant trait, all the individuals. The case second is that if we have got an individual with dominant phenotype, individual with dominant trait, now it is heterozygous. Then in the F1 generation, we'll get the offsprings, we'll get the progenies. In them, 50% would be having the dominant trait and 50% would be having the recessive trait. So the test cross ratio is 1:1 in case of the single character. So this was regarding the what is the test cross, what is the objective of the test cross. We'll see how we'll explain it with the help of a cross. So explanation. We know that Mendel crossed a pure tall pea plant with the dwarf pea plant, and in the F1 generation he obtained only tall plants. Now, to know whether the tall plant is pure, whether the tall plant which is produced in the F1 generation is homozygous or heterozygous, what the Mendel devised the test cross so as to determine whether the F1 generation individuals, whether the individual this homozygous, this individual with dominant trait, whether it is homozygous or heterozygous. So I will explain like this. Mendel devised a cross where he used it to determine whether the individual with dominant phenotype is homozygous or heterozygous. We know that Mendel crossed pure tall pea plant with pure tall pea plant with dwarf pea plant. So these are parents. Obviously this is phenotype. We have pure tall pea plant. This is male, this is female. We can say pure tall pea plant, it is being crossed with dwarf pea plants. And in the F1 generation, what he obtained? In the F1 generation, he obtained tall pea plants. What I mean to say is that Mendel crossed the tall pea plant, pure tall pea plant with the dwarf pea plant. And in the F1 generation, what he obtained? He obtained tall pea plants. But these tall pea plants, they would be having homozygous or heterozygous. That means these tall pea plants would be having capital T, small t allele combination. They would be having the genotype capital T, or they would have the genotype capital T, small t. Then how to determine that the individual with the dominant phenotype is homozygous or heterozygous? So Mendel devised test cross. Mendel crossed the pure tall pea plant with the dwarf pea plants. When he crossed the pure tall pea plant with the dwarf pea plant, he collected the seeds, then sowed the seeds in the soil. The field ultimately raised the F1 generation, first filial generation, individuals of the F1 generation. And what he observed that in the F1 generation, all the individuals in the F1 generation, they were tall. But he was not knowing whether the tall pea plants which were produced in the F1 generation, whether they are homozygous tall or heterozygous tall. That means whether they have genotype capital T or capital T small t. These individuals with dominant phenotype. So what he did, he devised test cross. He devised test cross. Test cross determines whether the individual with dominant phenotype, whether it is homozygous or heterozygous, whether it is having two identical alleles for the trait or two dissimilar alleles for the trait. So Mendel devised test cross to determine this. So we'll perform the test cross. We'll perform the test cross. So test cross in case of single character. In case of single character. So we have got test cross in case of single character. We have got parents. We have their phenotype is tall pea plant. It is pure tall pea plant. It is crossed with dwarf pea plant. Obviously, pure tall pea plant, it is crossed with dwarf pea plant. Then what would be the genotype? Genotype is, this is pure capital T. This is dwarf pea plant. It would be like this. Then gametes. Law of segregation will operate here. Two alleles of a gene will separate. This is one gamete. This is another gamete. This is one gamete. This is another gamete in case of dwarf pea plant. Then what is the F1 generation? F1 generation is first filial generation is will get this gamete, it will fuse with this gamete. That is what is called as, we call it random fertilization. Then we have fertilization of gametes. When there is a fertilization of gametes, then we get F1 generation. So this is F1 generation. This is capital T small t, capital T small t. Then this one and this one. This is capital T small t. This one and this one, capital T small t. This one and this one, capital T small t. So in the F1 generation, all this is tall pea plant. So this is tall pea plant. That means in the F1 generation we obtained only tall plants. Therefore we can say that the individual with dominant phenotype is homozygous for the trait, because we have obtained only the individuals with dominant trait in the F1 generation. Therefore we say that the parent with dominant phenotype is, individual with dominant phenotype is homozygous. Individual or parent with dominant phenotype is homozygous for the trait. Homozygous for trait. That is why it is homozygous for the trait, because we in the F1 generation we obtained only the dominant trait. We obtained the individuals with the dominant trait. We obtained the progenies with the dominant trait. We obtained the offsprings with the dominant trait. There was no recessive trait in the F1 generation. When there is no recessive trait in the F1 generation, we say that the individual with dominant phenotype, it is homozygous for the trait. So it would be obviously having capital T. Then this was case first. This is now we'll perform Case second is we have got test cross. So individual with, we have got parent. For example, we have got tall pea plant. We have got tall pea plant, but it is hybrid tall pea plant. It is crossed with dwarf pea plant. In test cross, we cross the individual with dominant phenotype with the homozygous recessive individual or homozygous recessive parent. So hybrid tall pea plant, it is phenotype. Then what would be the genotype? Hybrid tall pea plant, obviously the genotype is capital T, small t. Here the genotype is small t, small t. Gametes, capital T and small t. Here gamete, small t, small t. Then we will, there is the fertilization, what is called as random fertilization. And we will get F1 progeny of F1 generation, first filial generation. So obviously this gamete, it will fuse with this gamete. It would be like this. So capital T, small t. This is one individual capital. This another gamete will fuse with this gamete. So we have capital T, small t. Then this gamete will fuse with this gamete. We have small t, small t. And this gamete will fuse with this gamete. We have small t, small t. So in the F1 generation, we obtained two types of individuals. One is tall, and which have genotype capital T, small t, capital T, small t. That means phenotype is tall. Then these two individuals, small t, small t, small t, this is dwarf. That means it is 2:2. Two individuals are tall, two individuals are dwarf. So ultimately 1:1. So test cross ratio is 1:1 in case of test cross ratio is 1:1 in case of single character, because we have taken only one character, tall, into consideration, height of the pea plant, in case of single character. Why we write here single character? Because we have taken only one character, height of the pea plant, into consideration. Therefore the test cross ratio is 1:1. This is test cross ratio is 1:1. That means in case the individual is heterozygous for the dominant trait, then in the F1 generation we will get both the offspring is with dominant trait and offspring is with recessive trait. So in case of this type of cross, we have crossed a tall pea plant with the dwarf pea plant, and in the F1 generation we obtained tall pea plants as well as dwarf pea plants. That means the individual, the parent with dominant phenotype, that was what heterozygous for the trait. That means when the parent with dominant phenotype is heterozygous for the trait, then we get this type of ratio, that is test cross ratio is 1:1 in case. And it means that this ratio, here we have individual dominant phenotype is homozygous for the trait, but here we have obtained both tall and dwarf, tall as well as dwarf, in the ratio of 1:1. So individual, we can say that parent with dominant phenotype is, whether it is homozygous or heterozygous, it is heterozygous for the trait, heterozygous, and the genotype is capital T, small t, capital T, small t. So this determines the individual with dominant phenotype is homozygous or heterozygous. Then we have to devise a test cross for this individual, which will determine that the individual with dominant phenotype is whether it is homozygous or heterozygous. So it can be explained this. For example, if this is a tall pea plant, it's a tall pea plant, but we don't know whether it is homozygous or heterozygous. Then we have to devise a test cross for this individual with dominant phenotype. Then this was in case of single character, test cross ratio in case of single character is 1:1. Now in case of two characters, what is the test cross ratio in case of two characters? So we have got test cross ratio in case of two characters. We'll take two characters. For example, we'll take in case of single character, we have test cross ratio 1:1. This is 1:1. And in case of two, we'll take seed shape, seed color. We'll take two characteristics, seed shape and seed color, and we'll see their test cross ratio. Here in test cross ratio, in test cross where we take two characteristics, the individual heterozygote individual, that means we have capital R small r, capital Y small y, is crossed with homozygous recessive parent, So we have got this. We have heterozygote individual for two characters. We have got capital R small r, capital Y small y. It is crossed with what? Homozygous recessive parent. So it would be like this. And test cross in this case, the test cross ratio is for two characters. Test cross ratio for two characters is 1:1:1:1 for two characters. So for single character we have test cross ratio 1:1, and for the two characteristics, we have got test cross ratio 1:1:1:1. That means in case where we have to determine the test cross ratio for two characters, we have to cross double heterozygote individual. This double heterozygote individual is crossed with homozygous recessive parent. This is double heterozygote individual with homozygous recessive parent, and we will get the test cross ratio. So we have got 1:1 test cross ratio for the single character, 1:1:1:1 test cross ratio for this is test cross ratio for the two characteristics. This is important, that is test cross ratio in case of single character, in case of two characteristics, or in case of in case of one trait, in case of two traits. So we can say that so what in test cross, so finally we have completed what is test cross. In test cross, the individual with dominant phenotype, the parent with dominant phenotype, is crossed with homozygous recessive parent or homozygous recessive individual. And why this test cross is being performed? Test cross, it determines the zygosity of the parent, whether the parent is homozygous or heterozygous for the trait. So test cross is being performed to determine the zygosity of the parent parent, to determine the zygosity of parent, whether parent is homozygous or heterozygous. Is homozygous or heterozygous. Homozygous or heterozygous for the trait. So in test cross, the individual with dominant phenotype is crossed with homozygous recessive individual, and test cross determines the zygosity of the parent, that is whether the parent is homozygous or heterozygous for the trait. Hope you have understood. Thanks.